Na minha escola, cada aula me leva mais longe. Aprender lá me abre as portas do mundo todo, do Canadá para onde eu quiser ir. A gente aprende brincando.
A gente aprende investigando. We learn in 2 languages. We learn for life. That's the conclusion of my work. E a gente ama aprender onde quer que a gente esteja.
Na Maple Bear, o aprendizado é para a vida, com ensino multicultural que prepara seu filho para ser bem-sucedido em qualquer lugar, no Brasil e no exterior. Escola bilíngue com imersão em inglês e uma das melhores metodologias do mundo. Maple Bear, escola canadense de verdade.
Good evening, everyone. My name is Juliana Menezes, and I am the Chief of Projects and Operations Officer at Empathy, the Chamber of Commerce Brasil-Canadá, CCBC, in partnership with the University of Montreal, are pleased to present you the Tech Journey. This project brought together 5 master classes taught by renowned professors from the University of Montreal. Tonight, we will listen to Professor Nadia El Mabrouk. Dr. El Mabrouk is a professor at the University of Montreal and holds a PhD in theoretical computer science. Her research area is computational biology. Her main investigations are related to the development of algorithmic and mathematical methods from the perspective of comparative genomics. As a sponsor of this project, I would like to thank you all for attending, and with no further ado, I have the honor to call Dr. El Mabrouk, who will talk more about bioinformatics, its potential and implications for genetic processing. Dr. El Mabrouk, the stage is yours.
Okay, so thank you. So I have to share my screen. Okay, let me share the screen. Okay. Let me see. Okay. Okay, so hello everybody. So I'm not seeing anyone here. Okay. I have to. Okay, so thank you for the invitation. So are you hearing me correctly?
Yes.
Yes. Yes.
Yes.
Okay, so hello. So I am Nadia El Mabrouk, yes, from the Department of Computer Science at the University of Montreal. Alors, je suis Nadia El Mabrouk, je suis du département d'informatique et de recherche opérationnelle de l'Université de Montréal. So the name is in French, or the name of my department is in French, because as you know, the University of Montreal is a French-speaking university, so I am also French-speaking. Alors, si à la fin, si certains parmi vous parlez français, alors sentez-vous à l'aise à la fin de me répondre, de me poser des questions en français. So, but don't worry, my presentation will be in English. Okay, so I'm going to talk about bioinformatics for genome and gene editing. So I would like to tell you how the new field of bioinformatics has allowed an incredible advances in understanding of genetics and also of the human genomes. Okay, so here I will take an example, which is the CRISPR-Cas technology. Okay, so so I'll take it as an example to illustrate all what bioinformatics is able, okay, to bring of possibilities. Okay, so CRISPR-Cas9 is a powerful tool for editing genomes that opened the door to a new generation of treatment of genetic diseases, for example. And bioinformatics has played an essential role in all stages of detection, analysis, classification of the CRISPR-Cas system. So as I told you, as I will use this example to draw a picture of the main advances and tools of bioinformatics. So the algorithmic tools for gene prediction, sequence alignment, motif finding, also phylogenetic inference. So my presentation will be in 3 parts. In the first part, we'll present this CRISPR-Cas system. So if you don't understand everything, if you are missing some basics in molecular biology, don't worry, because after that, I will present a brief review in molecular biology. So recall what DNA, RNA, proteins are. Okay, so and then I will present the bioinformatic tools that are the basis of the main softwares that have been developed for the CRISPR-Cas systems, but also for many studies okay, in biochemistry, in molecular biology. Okay, so let's begin with the first part. Okay, so CRISPR-Cas, what is it? So it's a tool, it's a powerful tool for editing genomes. It is adapted from the natural defense mechanism of bacteria, who use it as a means of defense against viruses. So during a first viral attack, the bacteria store segments from the virus in their own DNA, and then this segment is used to recognize subsequent viral attacks and cut the DNA to eliminate the virus. Okay, so it would have not been so interesting if we couldn't adapt it to the human. Okay, but the fact is that we can use it also to edit human genes too. Okay, so actually the 2020 Nobel Prize in Chemistry was awarded to these 2 ladies, Jennifer Doudna, a professor at the University of California, and Emmanuel Charpentier, director of the Max Planck Unit for the Science of Pathogens. Okay, so they have been awarded for their work to adapt this bacterial system into a practical gene editing tool for humans, of course. Okay, so CRISPR-Cas is a revolutionary tool for biomedical research. Okay, so it allows changing, inserting, deleting sequences from the DNA and modifying gene function. So it can be used to engineer cells, tissues, and organisms. It can also be used to correct genetic defects, treat and prevent the spread of disease, and also it can be used to improve the growth and resilience of crops. Okay, so it's a very important tool, which is new actually, but it's not the only tool for editing genomes. There have been other tools before, but actually it's much easier than previous methods. That's what makes it important. It's much easier than previous methods for genomic manipulation, as these methods are limited because, okay, I have to move this window. Okay. Because they need to engineer a specific protein for each DNA site, okay? While CRISPR it doesn't need to have one protein for each site. So I will explain, you will understand better after that. Maybe if I can make this window smaller, because I can't see my slides. Okay, so CRISPR, so what is it? It naturally functions as adaptive immunity mechanism in bacteria. So that's what I told you. So what makes it interesting is that it can be transferred into other more complex organisms, such as eukaryotic cells and human cells, and can be used to edit genes. Okay, so the Cas protein actually is an enzyme that cuts foreign DNA. The protein binds to some RNAs, which together guide the Cas9 enzyme to a target site in the DNA strand where the protein will make its cut. Okay, so George Church here from Harvard Medical School expresses it simply this way. So operationally, you design a stretch of 20 base pair, that match a gene that you want to edit. And from there, one figure out what the complementary CRISPR RNA sequence would be. Then the RNA plus the protein will cut like a pair of scissors, the DNA at that site, and ideally nowhere else. Okay, so it's as simple. Yeah, you take a stretch of 20 bases from a gene that you want to edit, and you incorporate this stretch in a complex, in a Cas9 and RNA complex, and that makes the job, okay, of editing the gene. Okay, so then once the DNA is cut, the break is repaired by filling into gap with a sequence of nucleotides, and scientists can provide the DNA template of their choice, allowing to insert a gene or correct a mutation. Okay, so what CRISPR means, the term CRISPR stands for clusters of regularly interspersed short palindromic repeats. Okay, let me. Okay, and I will show you, so maybe you will better understand with this small video. So here is a small animation to summarize like what I just explained.
When bacteria survive a viral attack, they incorporate snippets of the virus's DNA into their own genomes. Those stolen segments and the genetic padding between them are called clustered regularly interspaced short palindromic repeats, or CRISPR. If the virus attacks again, the bacteria use those CRISPR segments as a template to create strands of RNA that home in on the corresponding sequence in the virus's genome. The CRISPR RNA carries along a protein called Cas9 to the target location on the DNA. The protein disarms the virus by cutting its DNA at that spot.
No, sorry. Okay. So the CRISPR-Cas system is a part, is a sequence in the genome, in the bacterial genome, which is composed of 3 parts. Okay, the first part is an array of repeats separated by unique sequences called spacers here, you see. So it's a sequence of repeats. The bold these blue here are repeats. Repeats mean sequences that are exactly the same. Okay, so we will see later. I will show you sequences, how it looks like. Okay, so there are repeats here, and then in the so separated by spacers. So the spacers are these sequences coming from the virus. Okay, so these stolen viral sequences that are inserted between two repeats. Okay, so the first part is an array of repeats separated by unique sequences called spacers. The second part is a leader sequence, abstract here, this one here. No, I have, okay, this one. So actually it contains the promoter. The promoter is a sequence of DNA which is needed to initiate the transcription, which means the expression of the genomic region. And third, we have an operon, so a region with genes, with the name of the genes are Cas, so many genes, many Cas genes. So depending on the type of the CRISPR-Cas, we have different genes, and I will show you later also how they look like. Okay, so this CRISPR-Cas system has three functions. The first function is adaptation, so where a fragment, as I told you, as you have seen, of a foreign genetic material, a virus, okay, is selected and incorporated in the CRISPR array as a new spacer. Okay, so a new repeat and a new spacer is inserted in the array. So that means that the spacer arrangements reflects the infection history of the host, and we can even reconstruct okay, the order of the infections that the host have been affected to. Okay. Okay, so the second function is expression, where the CRISPR array is transcribed and processed into mature CRISPR RNA that consists of single spacers. Okay, so the repeats here and the spacers are expressed in this type of structure. So a spacer and the repeat, which forms a strand here and a loop. Okay, actually, because it's an RNA, and also I will show you later how they looks like better than that. Okay, and the third function is interference, where the mature CRISPR RNA bound to the Cas9 enzyme recognizes and cleaves the corresponding DNA. Okay, so actually there is a variety of CRISPR systems requiring various types of Cas genes. What is interesting with the type 2 CRISPR systems is that it requires a single enzyme, Cas9, to catalyze DNA cleavage. So that's what makes it interesting, because we just need to incorporate one enzyme in the human genome, let's say, in the human cell, to make it work. Okay, so engineering the human genome is made simple as it requires integrating a single protein, which can be programmed to cut the DNA at any site guided by a given RNA sequence. Okay, bioinformatics. Okay, so now so this is the discovery, you know, the very important discovery for many applications. Okay, so now I would like to use this example to show you how bioinformatics have been important, how computational biology have been important in all the steps of the detection, analysis, classification of this CRISPR-Cas system. Okay, so that's it. So the bioinformatic methods, softwares, and tools used for the discovery and analysis of the CRISPR-Cas system are very well reviewed in this paper by a bioinformatics group in Germany. So I based a little bit my presentation. Okay, so this presentation, I used it for my presentation actually. Okay. Okay, so the main, so actually, so this is a figure from the paper. So the main software used for the identification of, okay, so the main software used for the identification of the genes, of the Cas genes, the identification of the array of repeat and spacers, and also the identification of the leader sequence are all given in this diagram, summarized in this diagram. Okay, so they are specifically, I will not right now, I will not present you each one of these softwares. Okay, it would be much too complicated. Maybe one presentation would be needed for each of these tools. But I will present the basics, the basic tools of bioinformatics that are used in all these ones. Okay, so these ones are specifically designed for the characteristics of the CRISPR's arrays and Cas genes, but at the same time, they require the use of standard bioinformatic tools, such as, for example, the BLAST-based protein homology search tools, pattern matching tools, pairwise and multiple alignment tools, phylogenetic reconstruction tools. So, and I will talk about all these in the third part of my presentation. So just an introduction to these main tasks in bioinformatics. Okay, so that will be my goal to show you so how it works. What are the basic tools that are used actually in almost all pipelines in bioinformatics. So anyway, to help biologists discover, annotate, study the sequences, the new sequences that they have. Okay. But before that, I need to take a step back and introduce the basic concept of molecular biology. Okay, so that I was talking about, but without explanation. So I talked about DNA, RNA, etc. So now I will just review what all that means. So I don't know if you have a good knowledge in molecular biology, you will not need this part, but I don't know about your background. So let's remind some basic notion of molecular biology. So this is my second part. Okay, so the central dogma of molecular biology. Okay, so the DNA is the DNA. It's for desoxyribonucleic acid. It's the medium that encodes the information that allow a cell and an organism to function and reproduce. So it is actually the basis of heredity. So each cell contains the same genetic code, but of course this code is not expressed in the same way in all tissues, okay? So, and the central dogma of molecular biology is that the process of extracting information from this DNA to make active molecule, this process is the same process among all living organisms. So the source is the DNA, and then DNA is transcribed into RNA, which is an executable code. Actually, it's transcribed into messenger RNAs, but also in other functional RNAs. And the messenger RNA is translated into the proteins which make the cell functions properly. Okay, so the DNA. So the DNA. Okay, so I guess you know about it, but let's remind it. So the DNA is a double helix of two polynucleotide strands bound together. So you see here the double helix with two strands. Okay, so the first strand, if you read it, you can make a sequence of letters, and the letters are A for adenine, C for cytosine, J for guanine, and T for thymine. Okay, so there are 4 nucleotides. So the DNA can be seen as a sequence on a 4-letter alphabet. Okay, so if you take one of these strands here, you can open it, and it gives you a sequence. So, for example, this is a DNA sequence. Okay, and the two sequences are bound together but by making base pairings. So the base pairings are between the nucleotide A and T and G and C. Okay, so these two strands are bound together with this Watson-Crick base pairings. Okay, and you have to know that in the human genome, for example, okay, so the DNA the genome. Okay, so this is the DNA. What about the genome? Okay, so each cell, each nucleus of a cell contains a genome, and a genome is a set of chromosomes, each being a separate double helix of DNA. So, for example, here, so this chromosome here is a double helix, okay? And the human genomes contains 23 pairs of chromosomes. Why pairs? Because the human genome is a diploid genome. So each chromosome is present twice, okay? And each chromosome is a double helix of DNA. So each cell in the body contains all these chromosomes. Okay, so this is about DNA. What about RNA? So RNA is for ribonucleic acid, okay? So the RNA can be seen, the primary structure of the RNA is also a sequence of 4 letters, but the T becomes a U. So the 4 ribonucleotides are adenine, cytosine, guanine, and uracil. So the thymine in DNA becomes uracil in RNA. Okay, actually the DNA is transcribed. So when transcriptions take place, okay, so let's say this part here is the gene which is expressed in the cell. So to do that, so the double helix opens, and then each nucleotide, bounds. Each ribonucleotide comes to bound with the corresponding nucleotide in the DNA and form the ribonucleic chain. Okay, so when you have a T, okay. So TC, okay, so this sequence of DNA makes this sequence of RNA, okay? So this is the second step. So the transcriptions make RNA from DNA. Now this RNA sequence comes, it's not double-stranded, it's one strand, but it folds. So it folds into structures. It folds by forming base pairing. Usually the base pairings are Watson-Crick and Wabble. Okay, so the most common base pairings are this one, A with U. Okay, A is paired with U, C with G, and also sometimes we can observe the pair GU. So for example, here you see from this sequence CACG, okay, this sequence falls to make this secondary structure here. Okay, so what I want, so this you had a few slides before a representation of this type, you know. They were actually the repeats of the Cas system, of the Crispr system. You know, the repeats are actually RNAs, and they fold in such a structure like this. So I will see them better later in the presentation. Okay, so to summarize, so this is a cell. So the DNA, which is in the nucleus of the cell, is transcribed into RNA. Actually, not the whole DNA is transcribed. The genes, some part of the DNA, okay, the coding part, okay, are transcribed into RNA, messenger RNA. And then the messenger RNA migrate to the cytoplasm of the cell, and it's translated into a chain of amino acids, actually into proteins. So into proteins. What are proteins? They are chains of amino acids, okay? Okay, so the proteins are it's the third important, very important macromolecule. Okay, so the proteins are macromolecule of amino acids, okay? And there are 20 or sometimes 22 amino acid, 2 rare amino acid, let's say. So a protein, a primary sequence of a protein here. If you see here, this is a primary sequence of a protein. It's a sequence on an alphabet of 20 characters. Okay, so here, for example, is an example of a protein sequence. So proteins are the macromolecule that ensure the functioning of a cell. They perform a large variety of functions within the organism, such as catalyzing metabolic reactions, play roles in DNA replication, in the cell structure, in transporting molecules from one location to another, or they play a role also in DNA cleavage, which is the case of the Cas genes, for example. The Cas9 is a protein, is an enzyme which cleaves the DNA. Okay, so the protein differs one from the other primarily in their sequence of amino acids. Okay, but this sequence actually fold, and it's the folding, it's the structure of the proteins which determines this its activity. Okay, so what is important, and an important field of bioinformatics also is to predict the structure of RNA, which fold also in 3 dimensions, but also of proteins, because it's actually the structures that determine the function. But to do that, we need the sequence. So we need to predict the structure from the sequence. Okay, so which are the parts that are transcribed and translated? These are the genes. So the genes are the part, are specific segments of the DNA along the chromosome that code for proteins, but not only proteins, also for functional RNA, for example. Okay, so we have, okay, so this, for example, and actually, so the genes can appear, you know, remember, so the DNA is a double helix. So, and the gene, so the sequence in one, you know, the sequence is not the same in one and the other of the, they are complementary in the 2 strands. And actually the sequence is read in the 5' 3' direction. So this strand is read in this direction, and the second strand is read in the other direction. So it doesn't make, it's not the same thing. We have to know whether the gene comes from is expressed in the first strand or on the other strand. Okay. Okay, so the genotype, which is the nucleotide structure of genes. Okay, so the sequence of the genes are the genotypes, and this genotype induces a phenotype. For example, the gene which is responsible for eye colors may lead to green or black eyes. Okay, so there are genes that are responsible for the color of the eyes. Okay, so depending on which allele do you have, so you will have red, a green or black eyes. Okay, so now the genetic code. So how to go from the DNA, which is a sequence of four letters, to the proteins, which are chains of 20 amino acids. So how to go from an alphabet of 4 letters to an alphabet of 20 letters. Okay, so recall that, so a gene here, so the DNA, a gene is a part of a DNA which is expressed, which is, let's say, this is the gene. This gene is transcribed into RNA, and then the RNA, it's translated into protein. Actually, the sequence is read, each triplet corresponds to an amino acid, okay? So it's read by triplets, by windows of 3 nucleotides. So for example, AUG correspond to a met amino acid, CCG correspond to a proline amino acid, GUU correspond to a valine amino acid, and that's the way how a sequence of RNA give rise to a sequence of amino acid. So this translation between codons and amino acid is actually made, it's the genetic code, okay? So the genetic code determines the correspondence between triplets and amino acids, okay? So this is the universal genetic code. Actually, now we know that the genetic code is not as universal as we thought at the beginning. Many organisms have slight differences in, okay, it's not actually completely universal, but this is the more frequent, the most used genetic code, let's say, okay? So, for example, so how you read it here? So UCC is a serine amino acid, okay? So if you take, for an alphabet of 4 letters, okay, each triplets can, okay, how many triplets do you have? You have actually 64 triplets. So you can make 64 triplets with 4 letters. But actually there are only 20 amino acids. So how does it work? It works because many triplets actually code for the same amino acid. For example, all these codons here correspond to the proline, to just one amino acid. So we say that the genetic code is degenerate. Okay, which means that many codons actually code for the same amino acid. Okay, so these were my short review on the basics of molecular biology. Okay, so let's now go to the bioinformatics part. I'm not seeing no. Okay. So now that we have reviewed the main concepts of molecular biology, we can see how the relatively new field of bioinformatics allowed for breakthrough in our understanding of the biological mechanism encoded into the DNA. Okay, so bioinformatics or computational biology, it's a field, it's a subfield of computer science. So it's a multidisciplinary field between biochemistry and computer science and genetics. It appeared in the 1960s after biologists discovered how to sequence DNA and proteins. So it's a field that appeared once the first DNA and protein sequence have been sequenced, okay? So from this sequence of alphabets, so the problem was to know how to interpret them, how to analyze them, how to find, how to understand the way they are translated into protein, etc. Okay. Okay, so and I guess you may imagine that with the new methods, the new sequencing method, okay, so there is an exponential increase of genetic sequences in the databases. Okay, so for example, this figure here represents the increase in full eukaryotic genomes appearing in the databases, but you have other figures like that for the number of protein sequences, etc. So it's an exponential. You have so the increase in the database is spectacular actually. So of course we can't analyze them by hand. So we need automated tools which are sufficiently efficient in speed and space. And so this gives a plethora of computational and algorithmic problems that have to be analyzed and resolved and to help analyze all this data. Okay, so let's say the first main problem okay, so let's say to have a sequence, we have to sequence it, but sequencing is not a bioinformatics problems. It's more biochemistry problem. Okay, but actually after sequencing, okay, we also need to assemble the sequences. Assembling fragments of sequences it's also a problem that needs algorithmic tools. But okay, and then when we have the sequence, when we have a sequence, for example, the human genome, then the problem is to know how to annotate it, what it corresponds to. So, for example, you end up with a sequence like this, and then you have to know what is it. Is it a coding sequence? Is it a non-coding sequence? Does it contain genes? Has this sequence already been completely or partly deposited in the database? So all these require algorithmic tools, you know. So all these are actually it's about analyzing a text. Okay, okay. And also, so after sequencing and assembling, the question is to annotate the genome, which means discovering the genes coding for the proteins and for the functional RNA and for other elements. For example, how to identify the CRISPR-Cas systems in such sequences. So in this letter case, this means finding the repeats, the leader sequence, the Cas genes. Okay, so we have, so these are the kind of problems that of course we face as soon as we have sequenced a new genome. Okay, so how identifying genes? So if we have no previous information on a related genome, then the ab initio gene prediction method rely on finding some specific motifs or on some statistical property of the protein coding sequence itself. So it's more difficult in eukaryotes because because in eukaryotes, the genes are made of exons and interspersed by introns. Introns are are non-coding sequences. So anyway, so it's let's say more difficult in eukaryotes. But in prokaryotes, such as bacteria, for example, genes are generally without introns, and annotating the genomes can be made by identifying open reading frame. What is an open reading frame? It's a sequence of a size exceeding a certain threshold, so it shouldn't be too short, okay? Which start with a start codon and ends with a stop codon and contains only triplets coding for amino acids, okay? So that's it. So you have to notice that when you have a sequence, you have to consider 3 possible reading frames in each direction. So a sequence, if it's coding for something, so it can, okay, you can read it in 3 ways, in one strand or the other. So for example, here, if you have this sequence here, so you can, if you begin with the G here, so if you read it from the beginning, it gives you a subdivision into triplet, beginning with GUC, which is a valine. But actually, if the beginning of the sequence is here, so then the amino acid would be serine from UCA. And now if you begin it at the 3rd position here, the first amino acid of the protein chain will be a histidine. So it's different. So all these, actually we have to analyze 6 different reading frame, 3 on one strand and 3 on the complementary strands. And usually only 1 reading frame is used for the translation of a gene. Okay, so that it's just to give you an idea of how we can identify the coding regions in a gene. Okay. So, but most frequently, genes are not identifying from nothing, you know. We don't, when we sequence a new genome, we know many things about closely related genomes. Okay, so actually what we do is to see whether we can import an information on a neighboring genome on the new genome. So most frequently genes are identified through sequence similarity. Okay, so for example, is a given, you have a sequence, is it a gene? So if you find the same, if this sequence is similar to a gene which exists already in another organism, then chances are that the sequence corresponds to a homologous gene. So when you don't know, you try to look for it in your database, or I don't know, you compare it with the human genome with the mouse genome. If you find the same sequence or a sequence with a high similarity, and if you know the function that the gene codes for a given gene, in the mouse, then probably it encodes for a related gene in human. So sequence homology between protein or DNA sequence actually is similarly defined in terms of shared ancestry. Okay, so homology among proteins or DNA is inferred from their sequence. Okay, so when 2 genes are homologs, means they descend from a common ancestor. So when we are asking the question of similarity, we ask the question of whether these 2 sequences have a common ancestor. So as we have no direct, we don't know about the history, you know. we don't have a direct access on evolution. Okay, what we have are the sequences that we observe on the actual species. Okay, so we can just infer the evolution on the basis of the comparison between the two sequences. So for example, here, if we have these two sequences, CAT, AG, TG, and the seconds here, so what we want to know is whether they are similar or not. Okay, so when we similarity, usually we calculate it. We count the number of nucleotides that are the number of mismatches. For example, here you see the A is substituted into a G, so there is a mismatch here. Okay, here the C is aligned with a gap, which means that the C in this sequence maybe have disappeared in the second here, or symmetrically it appeared in this sequence. Okay, so the similarity consists of finding maximizing the occurrence between the nucleotide in the two sequences. Okay, finding the best, so the edit distance, for example, between two sequences is the minimum number of mismatches, insertion, deletion that can explain how to go from one sequence to another. Okay, and so finding the edit distance between two sequences is something that I teach in the first classes, in the first hours of my bioinformatics course, for example. Okay, so this is done classically using dynamic programming algorithms, so this table here show you a dynamic programming table. So it's an algorithm that takes a quadratic time to find the best alignment between two sequences. So these are for nucleotide sequences. What about amino acids? So you can also align the amino acid sequences. So the problem is to know, but okay, so is to know which score you have to give to a substitution between two amino acids, because the probability of one amino acid mutated to another is different depending on the pair of amino acids. So for example, the Blossom table is a table of values reflecting the probability of a substitution of a mutation between two amino acids. And so these tables are used to compute the best alignment between two sequences. So it's a little bit difficult to explain in few minutes, you know. So this is like a few courses in a few hours in my course of bioinformatics. Okay. So these are actually the basic, so the basic problems in bioinformatics is to align sequences, because evolution works by duplicating and mutating. So many things in biology, a lot of things can be understand by comparing, by studying the evolution of the sequences. Okay, so it's a main thing to know how to align sequences. Okay, so when for example you have two sequences and you want to see if they belong to the same family, if they have the same function, I don't know. So you are interested in aligning them on their entire length. Or alternatively, if you have two sequences and you don't know if they are similar, but maybe they may contain one part in common. So in that case, you are making a local alignment. You are looking for the best match between part of the two sequences. Or if you have a gene and you have a sequence of a gene, and you want to look for it to find all occurrences of this this sequence in the genome, then you make a motif search. So you are looking for all the occurrences of the gene in the sequence. Okay, so all these problems can be computed, can be addressed using the dynamic programming algorithm that I have just gave you an idea about in the previous slide. Okay, but these dynamic programming algorithms, as I told you, are quadratic in time and space. And when you have huge databases, you can't use a quadratic algorithm to search for a sequence. So you need to have much faster algorithms. So that's, okay, so to do one of the most used motif searching tool is BLAST. So there are different versions for the BLAST tool for nucleotide or amino acid sequences. When you search for the gene, when you search for a sequence in a DNA or in a protein database. So BLAST, the dynamic programming algorithm is an exact algorithm, meaning that you are ensured to find exactly, for example, if you ask for the best for all the occurrences with 3 mismatches, you will find them all. But the BLAST algorithm is not an exact method, rather it's a heuristic, meaning that it is not guaranteed to find all the occurrences of a query sequence in a target genome, but it's much faster than exact dynamic programming method. And it comes also with statistical significance for output sequences. So the heuristic is based on a filtering method. So in the first step, so as you see it here, so all the window of a given size K of the query sequence are identified, which allow, okay, so you know, so you have a sequence that you want to find in your genome. So all windows K, all windows of size K, you look for, okay, you do an exact match of them in the sequence. So you identify okay. So all windows of a given size K of the query sequence are identified, and this allows to filter the database and to keep only the region that are likely to contain the query, okay? Then these identified seeds, K-mers that we call seeds, here are extended on the left and on the right, and also merged together to form larger sequences. So these algorithms depend on a set of parameters. For example, the size K of the seeds, the threshold under which we stop the extension, the minimum score of an output, and etc. Okay, so let's take an example. No, we'll not take an example. So, no, it's not an example. Okay, so so the previous slides were about how to identify some sequence. For example, if you are looking for here, for example, you are looking for Cas to come back to our CRISPR system. Okay, so Cas2 is one of the Cas genes. So let's say you have identified a sequence of a Cas2 gene in one species, and you want to look for them to look for them in other species. So you need, for example, to use a BLAST search tool, and then you identify the Cas gene is in these, for example, 5 different bacterial genomes. So once you have identified these sequences, so of a given gene family, the challenge then is to characterize the gene family to infer the evolution of the sequences to predict the protein structure, for example. So the second standard task in bioinformatics is thus to obtain a multiple alignment of the set of sequences. Okay, so here, for example, the same sequences of the Cas2 genes are aligned. Okay, so the goal is to find the best multiple alignment of all these sequences. And from this multiple alignment, for example, you see that at this position, you have almost always M's. So this is highlighted here in the signature of the family. So you have some signature. So this motif here represents represent this family of genes. Okay, it represents it's a short way to represent the alignment. So it says, for example, that for this family of genes, at this position, you always have these. Okay, at this position, you have two possibility of amino acid, or however, this position here is much more variable. Okay, so from a set of sequences, then you get a multiple alignment. The multiple alignment give you a characterization, okay, a profile of the gene families. Also from this alignment, you can infer the evolution of the genes. Okay, so the evolution of orthologous genes, and this tree gives you a rich source of information to understand the adaptation mechanisms and function of the genes. Okay, so this is an important part also of the process, is to infer the evolution, to know that. So when you infer this, you know that this gene is closer to this one, and this orthology information gives you a lot of information about also the evolution and the function of these genes in different bacteria. Okay. Okay, I don't know how much time I have right now actually remaining. Maybe I can skip this one. So this is a multiple alignment algorithm. So you can have many different, there are many different multiple alignment algorithm. One, the most used one maybe is clustal W, but maybe I will skip this slide here. Here is, okay, to go back to my CRISPR system. So these pictures show you how the repeated sequences and spacers looks like. So recall that so this is the CRISPR array. Okay, so remember, it's so the array here is a sequence of repeats interspersed with spacers. So the repeats are sequences that are identical or almost identical, and you see them here. Okay, you see, so it's like this one would be this sequence here, and this is the spacer, and the second would be this one. Actually, all this, okay, this is the consensus sequence of repeats, which means that the blue parts here are this sequence here, okay, with a very small variation. So, for example, let's say this repeat here will have an A in place of a G here, but they are all the same in the other positions. Okay. So this here is how the repeats look like, and recall also that the spacer should be all different, because the spacer are subsequences that comes for different viruses. So there is no reason they are similar. Okay, so actually they are not similar. So these are the spacers, and you see that they are not similar. Okay, so so in the figure that I showed you in the beginning, one of the program used to identify to predict the CRISPR arrays was CRISPR Finder. Okay, So CRISPR, actually the detection of CRISPR arrays stands on, so detecting this array stands on detecting identical sequences flanked with all different sequences. Many computational methods have been designed, some of them based on searching with a blast search, for example, a set of known repeats. Others, such as CRISPR Finder, is using more sophisticated pattern matching techniques based, for example, on suffix trees and suffix arrays. So these are data structures allowing to represent the internal structure of a database, leading to time-efficient exact search. Okay, so it is especially well designed for finding repeated sequences in a text. So for example, here you have the input sequence. So this is an illustration of the CRISPR Finder method. So you have your sequence here. First, you identify repeats using a suffix array. So the same color parts are the repeats. So for example, the yellow part here corresponds to the yellow part here. The blue parts correspond to each other. Okay, so these repeats are identified using this sophisticated data structure. Then there is a filtering of the candidate, removing the two large repeats, because we have an idea about the maximum length of a repeat. So there is a filtering here. Then repeats are clustered to form arrays. Okay. And then we discard tandem repeats, which means that, okay, we discard tandem repeats, and also finally to check that each part here corresponds actually to a CRISPR array, the sequence, the spacers here should be different. So they have to, so their similarity shouldn't be greater than 60%, for example. So the last step is about checking whether the spacers are sufficiently different to correspond to actual spacers. Okay, so this, for example, what is done for predicting the CRISPR arrays. Okay, so I recall you too that the repeats are actually RNA sequences which fold into structures. So here are, for example, two structures of two known repeats. Okay, so you see that they fold into some structures. And predicting actually the folding of a given RNA based on its primary structure is another well-studied field in bioinformatics. So when you have the sequence, how to predict the structure is also a very important and classical problem in bioinformatics, and it's also one part of my course in bioinformatics, for example. Okay, and I think I have to skip the phylogenetic part of my presentation. Let's say that, you know, I have showed you one tree. So these were phylogenetic trees. So the phylogeny intend to find the ancestral relations between sequences. Of course, the main goal of phylogeny is to infer the evolution of species, and we infer it from the sequences, actually, not from the genome, not for the complete genomes. So to study the evolution of the species, we need to find nice footprints of the evolution and to align them. And you have many different phylogenetic tree inference methods. You have the distance method. So I think I will skip all this. You have the maximum parsimony or probabilistic methods to infer phylogenetic trees. And it's not an easy problem because the space of trees is huge. So you can't explore all the trees. So you have to find strategies, heuristics to explore the space of trees. Okay, and finally, you can also, so this is for one gene, but when you look at the genome, you can represent a genome as as a sequence of genes too. And for example, when you compare two related species, you can find the same genes, but not in the same order. So for example, here is a picture comparing the human and mouse genomes. So you see the mouse genome, the colors correspond to the mouse genomes, to the mouse chromosomes, okay? And then the colors are reported to the human chromosomes, okay? So what you see here are the human chromosomes. That means that in the chromosome 1, for example, of human contains part of the chromosome 1 of mouse and the chromosome 2 of mouse and the 4, you see? So you have a mixture, you find the same content but in a different order. And then very important problems, which is actually my expertise, that's the field I'm working on, is to predict, to infer the global evolutionary event that have shaped the organ and also organizations of genes in the genomes. This is for to come back to the CRISPR-Cas system. Actually, the Cas, these Cas genes, you see, before the leader sequence, you have a sequence of Cas genes. So it represents, it's a centenary, it's a sequence of genes, and they are not exactly the same in different species and not exactly in the same order. And it's important to understand the function of of the different type of CRISPR-Cas. It's important to know how to classify them correctly, and to do that, you have to infer the evolution of this organization of genes, how they have evolved through duplication, losses, horizontal gene transfers. And this is something we are working on my lab. And Matteo Delabre, my PhD students, who helped me preparing this presentation, is actually working on this problem, trying to infer the horizontal transfers. So the transfer from one bacteria to another, okay? So the movement of genes from one bacteria to another that can explain this organization. Okay, so this is my the end of my presentation. So to conclude, Bioinformatics has made possible unprecedented discoveries in genomics and molecular biology, given the huge amount of biological data accumulated in the databases. The development of automatic tools for the analysis, classification, prediction, and discovery of new function is absolutely essential. So I have used the CRISPR-Cas example to illustrate how the classical bioinformatic tools were crucial to unravel the complex function and evolution of CRISPR systems. Now, the field of bioinformatics evolves with the new problems encountered in biology. For the CRISPR-Cas, for example, the identification of anti-CRISPR proteins involved in the inactivation of mechanism of the CRISPR-Cas is one of the main bioinformatic challenges for the future. Okay, and thank you. And then they send me some slides that I have to present on the University of Montreal. So I made them in French because, you know, because you have to know that the University of Montreal is, first of all, a French-speaking university. Montreal is in Quebec, which is a French-speaking city and also province. Okay, so welcome to the University of Montreal. So this is a short description. So you see, we have, okay, so this is, donc on existe depuis 140 ans. So this is okay, so this is the time, donc l'Université de Montréal existe depuis 140 ans. This is the number of students in the university. This is the number of professors. This is the number of active people active in the world that are coming from the University of Montreal. The University of Montreal is the 4th university in Canada in the volume of research activities, etc. Okay, so it's a nice university, and Montreal is a nice city. And here you can, okay, maybe the field that I was talking about is in this part here: biology, chemistry, mathematics, computer science, but you can study in any field that you want here. Okay. Voilà, and this is to tell you that the university, Montreal is a nice city that is worth visiting and coming to live in. And if you need information about the university, you can find many tools on the web. Okay, and all the questions about the admission at the university. Okay, so thank you very much, and I let you. So now I'll stop my sharing, I guess.
First of all, thank you so much, Dr. Mabrouk. It was a pleasure to have you sharing your knowledge with us tonight. And I think that we have time for a few questions from the audience. So let's start with the first one. It's from Gabriel de Souza. Is there any programming language preferred in bioinformatics, such as R or Python?
Yes, maybe. Yes, I would say Python, yes, but no, not everybody programming. Yes, Matteo, in which language do you program? Can you tell us?
Yeah, Python is the main language in bioinformatics today, I would say.
Perfect. So the second one is from Marcos Calais da Silva. What kinds of algorithms are in your field of vision right now? Are there models that use the pattern of expressed protein distributions of databases or experimental data, for example, for validation or refinement of traditional sequencing?
This is a very complicated question. So I'm sure there are a lot of tools, but I would not be able to say which one this question is asking about, but maybe you can tell it again.
Okay, so what kinds of algorithms are in your field of vision right now?
The field of vision?
Yes.
Okay.
Are there models that use the pattern of expressed protein distributions of databases or experimental data, for example, for validation or refinement of traditional sequencing?
Okay, you know, there are many models in visions, but I will not be able to answer to this question very easily like that. Okay.
Okay. And the last one, what are the contributions of bioinformatics in the study of SARS-CoV-2?
Yeah, that's a good question. This is a whole field. For example, in the ISMB conference, in the most important conference on bioinformatics, and there is a full day, you know, where about the discoveries in SARS-CoV. So yes, there is a huge amount of research of bioinformatics in this field too.
Okay, so now the last one.
No.
What drew you to the field of bioinformatics?
Pardon?
What drew you to the field of bioinformatics?
What drew me to the field of bioinformatics? Okay, so I did my PhD in the 1990. Okay, when I did my PhD, the field was actually beginning to arise, you know, and it was a new field that was interesting for people in pattern matching, because so my PhD is on theoretical computer science, so on pattern matching, so on studying, finding pattern matching is about studying, analyzing text, and the DNA, the proteins give a huge number of problems, because it's actually the problem of analyzing a text of words. So it's a very interesting application of all the algorithms that have been developed in the field of pattern matching. So I was in a lab in France with people, famous people known in the pattern matching field. So it was the beginning of bioinformatics, and it was a natural things to do, a natural new field to explore in computer science.
Great. Thank you so much, Dr. Nadia. Do you have any last words?
So thank you very much for this opportunity to talk to you in Brazil. Okay, so it makes us travel a little bit. And okay, so if I was able to speak Portuguese, I would have ended my presentation with few words in Portuguese, but I'm not. So that's why I ended with some. I would end with some words in French. Okay, alors merci beaucoup pour l'invitation et au plaisir de vous voir ici à Montréal.
Thank you so much. Maybe you can try to say obrigada.
Obrigada.
Yes, perfect. Thank you so much, Dr. Nadia. And now I'd like to call Juliana Menezes again to deliver the closing remarks.
Thank you so much, Dr. Mabrouk. Obrigada. It was a pleasure to have you with us tonight, sharing your knowledge. I am sure all the participants enjoy your masterclass as I did, and I learned a lot about bioinformatics and genetic processing. Thank you once again for accepting our invitation, and we hope you can learn more with you someday. Finally, I would like to give a special thanks to our sponsors, Air Canada, Maple Bear, CEO Lab, and Empathy. Once again, thank you all for attending tonight, and we hope to see you soon on Monday, 7 p.m. to learn more about the future of home office in the post-pandemic era. Thank you, everyone. Have a good evening.
Good evening.